Welcome to Yersinia-Omics, an interactive multi-omics database dedicated to Yersinia species. This platform centralizes over 300 genomes, 300 transcriptomes and more than 80 proteomic datasets, and makes them easily accessible through a set of browsers and viewers. Let's dive into how you can use it, beginning with the homepage. On the homepage, you will find shortcuts to the most commonly used reference strains, giving direct access to their gene viewers. From here you can enter the three main data browsers: Genomics, Transcriptomics, and Proteomics. Let's start with the Genomics browser. On the left, a phylogenetic tree built from core genes shows the relationships between the available Yersinia genomes. On the right, a searchable table allows to emphasize searched strains. Clicking on a strain also highlights it in the phylogenetic tree. The table lists key metadata for each assembly: species, lineage, isolation source, year, plasmid content, etc. Double-clicking on a strain opens its gene viewer for detailed exploration. The gene viewer is the central workspace of Yersinia-Omics, also accessible from the shortcut on the homepage, opening in a new tab. At the top of the page you can select other genomes, chromosome or plasmids, with external link to corresponding RefSeq assembly and downloadable annotations. On the left, a searchable list of annotated genes allows to select the gene of interest. Each gene page presents identifiers, genomic coordinates, functional annotation, sequence data, external link to GenBank protein and other annotations. For certain reference strains, Yersinia-Omics provides an optional Synteny view powered by SynTView. The Synteny is automatically centered on search gene. SynTView aligns homologous regions across multiple genomes, revealing conserved blocks and local rearrangements. You can also inspect protein homologs across all Yersinia genomes, a helpful tool to look for homologous locus in other reference strains and to inspect sequence conservation across Yersinia. You can search for specific text in the table and highlight homologs in the phylogenetic tree. BLASTP results such as coverage percentage, similarity percentage, or E-value are listed on the right of the table. Double-clicking on a homologous gene will open the gene viewer of the homolog on the other strain genome. The gene viewer also allows quick access to common external databases. The Kyoto Encyclopedia of Genes and Genomes, better known as KEG, lists annotation and pathway, such as metabolic pathways, related to the selected gene. KEG pathways can be opened, and the selected gene will appear highlighted. Here the FADB genes or locus YPO3766 is highlighted in the fatty acid degradation pathway. UniProt annotation can be browsed with quick access to Go terms, bibliography, chemical reactions, or structural information such as PDB or AlphaFold prediction. The Interpro website allows to browse annotated protein domains gathered from various databases, and to find homologous domains in other species. Intact shows experimentally validated interactome data. The string database is also available. Each database can be opened in a new tab of your web browser. Coming back to Yersinia-Omics internal data. The Transcript Differential Expression tab allows to find microarray or RNA sequencing experiments where your gene of interest is up or down regulated, after selecting a specific fold change and p-value cutoff. Each comparison name explains in detail the biological conditions that were compared in the experiment. You can select comparisons to be opened in the Genome Viewer, that we will see later. The Transcriptomes tab shows TPM normalized values of RNA sequencing experiments for the selected gene. The Protein Differential Abundances tab and the Proteomes tab work the same as for transcripts, but for proteomics datasets. Next, let's look at the Transcriptomics browser. After clicking on it from the homepage, it will open in a new tab of Yersinia-Omics. Each row represents a biological condition, described by biological condition metadata such as publication date, strain, temperature, medium, technology, bibliographical reference, or project number. Filters on the left allow to quickly select relevant conditions based on the metadata. Technology use such as microarray or RNA sequencing. Specific mutated strain, temperature growth, or growth medium such as LB or in vivo experiment. You can look for specific text in all the fields of the metadata. You can then select biological condition of interest and identify the relevant comparisons from the same study, based on the publication date, for example. As experimental conditions differs between studies, cross-comparison between studies are not available. After selecting conditions, experiment can be opened in the Genome Viewer. Here for RNA sequencing, the sequencing coverage is shown over the whole genome. Transcript direction is represented by the orange, positive, and blue, negative, colors. You can navigate the genome by right-clicking on the side of the window. Coverage data allows to observe transcripts on unannotated regions, 3' or 5' UTR regions, or antisense RNA. You can search specific region by gene or locus name. Here we see that the DKSA gene shows no coverage in the condition of the DKSA mutant, while we see transcript in the wild-type strain. You can switch from absolute expression to relative expression between two conditions. Comparison between the selected conditions and other conditions of the selected experiment will appear. From the transcriptomics browser, you can also open selected conditions in the heat map viewer. Comparisons with the selected conditions appears in columns, and each gene of the genome as rows. Hovering over a column header displays comparison name. Clicking on it allows to sort on the log2 fold change value. Loci appear in other columns, along with gene coordinate in the genome, strand, gene name and gene product. Let's focus on RNA sequencing analysis report, based on the Sequana pipeline. Searching for a specific study, here Aviken et al. From 2015, we can find some external link, such as the corresponding article or NCBI project number. The article can be opened in PubMed or bioRxiv. When available, you can click on the project number in the DSeq2 report column. It will open a new tab in uBrowser showing a HTML report from the Sequana pipeline analysis. Relevant comparisons of the whole study are listed, with the number of up and down regulated genes. Quality control plots are shown, such as total read count per sample, proportion of null counts, count density distribution, or most represented gene in each sample. Sample are sorted by hierarchical clustering to ensure replicability, or displayed on principal component analysis to capture replicate variability. An interactive 3D PCA is also available to better explore sample clustering. Hovering over the data points displays sample condition and position on the three principal components. Other normalization and distribution plots are also available. An interactive volcano plot allows to have an overview of differentially expressed genes for each comparison. Hovering over the data points display locus or gene name, log2 fold change significance and mean expression. For each comparison, the differential analysis results are displayed in a sortable and filterable table that can be copied or exported. From the home page, clicking on the Download Process Data button gives you access to a server which stores all the transcriptomes and proteomes data available on your Cineomics. After selecting your data type and strain of interest, table of expression, fold change, p-value or adjusted p-value data encompassing all the experiments done with this strain are available for download. You can save the table and open it on your computer or in data analysis software to perform your own analysis. As for the heat map viewer, the columns represent condition or comparison and gene metadata. All the genes from the selected strain genome are listed in the rows. For more information on your Cineomics usage, refer to our published article from 2023, and please don't forget to cite it if using our website. Don't hesitate to reach out if you want your own data to be added in the database. Thank you.